Mendel's monohybrid cross, or we also call it as one gene inheritance. It is a very important form of crossing what we are going to do, because it comes for 3 to 5 marks in PU board. In the year 2018 March also this question has come. So, in the slide you can see that we have asked a question, can you derive the phenotype and genotype ratio of F2 generation? So, we will see what we can do about it. So, any such question comes, the first thing you do is definition, you write definition. So, definition will give you introduction to the entire answer. So, what is the definition here? A cross involving two pure breeding parents differing in a pair of contrasting factors. So, I hope this statement is clear. We are going to cross two plants which are of pure breeding, meaning to tell it is homozygous, but they are differing only in a pair of contrasting factor. We will see how that happens. So, we are going to take a parent which is either completely tall or completely dwarf. So, this is tall and this is dwarf, right. So, this particular plant which is tall is homozygous dominant, and the other plant is homozygous recessive, right. So, we are having these two plants here, one is homozygous tall and the other is homozygous recessive. So, what we are going to do? We are going to cross between them. So, we are going to mate it. So, first what we have to do is we have to get the gametes. So, always remember gametes are haploid and you get single forms. So, I have got capital T and small t gametes which will be crossed, and I will be receiving a offspring. Offspring is otherwise called as filial. So, here it is the first generation. So, we call it as first filial generation. So, what I am going to do here in first filial generation, in a short form I write it as F1. So, I have got a hybrid in case of first filial generation. So, what I am going to do now? I will be doing a selfing. Next step what I am going to do? I am going to self capital T into capital T. So, I am going to self it. We call it as selfing. So, what I have done is first filial generation, two plants I am going to cross, because I know it is a tall plant, but I do not know what kind of tall, because if I see it genotypically I will get to know it is heterozygous tall, right. Okay, fine. Let us continue the answer. So, after I have self, that is capital T small t crossed with capital T small t, again I will be receiving gametes. So, this was my first generation, and so I will get gametes here again, where I will get two different types of gametes since I started off with heterozygous type. So, gametes here. Now, this could be crossed. I could cross directly or I could show it in Punnett checkerboard to get my second filial generation. So, let us draw a Punnett checkerboard. Always remember there should be one row and one column more than the number of types of gametes. So, here we have two types of gamete, that is capital T and small t. So, I have kept one more extra column. Similarly, here I have capital T small t. So, assuming one behaving like a male, the other behaving like a female, I start crossing. So, I take first T with this first T and I get capital T, capital T. Similarly, I cross the first row with the second column, I get capital T, small t. I am crossing second row with first column, again I get capital T, small t. And this one here I will get small t, small t. So, what I get is the second filial generation. So, let us break up and see how it is. So, we have two different ratios to find out, if you remember the question. One is genotype and the phenotypic ratio. So, one is genotypic ratio we are trying to find, the other is phenotypic ratio. Okay. So, genotypic ratio. So, if you see first gene, this looks different, capital T, capital T. So, what is this kind of thing? It is homozygous tall. And how many number of homozygous tall you could find in this Punnett checkerboard? 1. Then you have the second category as capital T, small t, and you can see 2 in number. So, you have 1 and 2. So, 2 capital T, small t. You have heterozygous tall. Then you have the third criteria. This is the last box here, small t, and it is also single in number. So, you have homozygous dwarf, right? Yeah. So, can I come to a conclusion that genotypic ratio is now 1:2:1, whereas let us see the phenotypic ratio. If you see, this particular plant is tall, this is also tall plant, this particular plant is also tall, but this is dwarf, right? So, I have 3 tall plants and 1 dwarf plant. So, what do you understand? Phenotypic is outer morphological factor and genotype is internal factor. Okay. Just let us see the marks distribution. Definition would carry 1 mark. The parental generation crossing will be 1 mark. First filial generation will be 1 mark. Okay. You have the genotype carrying 1 mark, and phenotype, which is 3:1, will be carrying 1 mark. So, totally it is 5 mark.